I will present ongoing work on patterns of protein expression on a model organism which is rather suitable for studies on adaptation and ecological speciation. This work is not so big or ambitious like the collaboration that we shared with Kerstin Johannesson, Roger Bartling, Jerry Coyne and others, but still within that we can incorporate some new insights on even in such a topic. In addition, this work has been done in collaboration with two other workers, a postdoc from my group, Ángel Pérez Diz, and Maria Paez de la Cadena, which is a biochemistry professor from our department. Thanks to Darwin, we have a nice and powerful mechanism which allow us to give an explanation for processes like adaptation and ecological speciation. Of course, there is some contentions about the relative importance of ecological speciation, but I think that most of us will agree that must be a very important factor, at least for animals with sexual reproduction. We have improved quite a lot our knowledge in relation to the ecological speciation process, but mostly our improvement has been produced on morphology, on ecological topics and similar, but our improvement has been not so large in relation to the molecular level. Of course, there are a few exceptions that can be used just to show that some genes are producing some particular effects related to the environmental and to adaptation, ecological speciation. But for most non-model organisms, we need to apply Ohmic technologies. Those Ohmic technologies try to scan in a massive way a number of genes or their products in order to find candidate genes of the adaptations process or the ecological speciation ones. Typically, most studies have been done in evolutionary area using massive scan methods on genomics and transcriptomics, but there are relatively little work made on the proteomic part. And this is one of my objectives today, trying to show you that there is relatively good reasons to attempt to these methods in the future for your model organisms. So why using proteomics? First, there are some methods of proteomics, not all of them, of course, that can be used in any species without any previous knowledge. Second, it's relatively simple, fast, and cheap, of course, if you don't apply mass spectrometry, which is quite expensive. It's closer to the molecular phenotype, and so to the phenotype level too. And interestingly, there is a very poor correlation between the level of transcriptomics and the proteomics for those model organisms which has been studied in detail. Theoretically, it could be possible to use for identifying candidate genes, but of course I must admit that this approach is more useful for model organisms and is more difficult for non-model ones. I came with a similar model organism, similar, not the same, than Kerstin presented yesterday. And so I will not go into the details because she already gave many of them. But I want to emphasize some difference of our system in northwest of Spain regarding other similar or related hybrid zones in Litorina. We have our ecotypes living in microallopatry or sympatry, as you want. It's a problem of definition. But one of the, following the Kerstin notation, crack-protected form is larger, living on the upper shell, on barnacles, and the wave-protected one, more or less related to the wave-protected of the others, also lives on the lower shore, and they are affected by very different ecological conditions and ecological needs. Interestingly, the two habitats overlap partially, and then you can find both forms together and a variable percentage of hybrid forms. And this is a quite strong situation in the Litorinid world, because there are not many places in the world in which you can find so easily, separated by a few meters, these so nice hybrid zones. The hybrid zone has another advantage in this species in the sense that you are allowed to get mating pairs and so estimate directly the sexual isolation and the sexual selection in the wild. And using these and similar methods in our group, we have found that the assortative mating is quite strong between the ecotypes at the midshore. It's nearly 70% of the maximum possible, suggesting that they are already evolving some kind of reproductive isolation between them. We have already a lot of work that suggests that those mechanisms contributing to this effect are in one way or another a byproduct of divergent adaptation. So I will say that scythe in this organism is another magic trick, and I love to answer questions about this topic, so you are welcome. Anyway, on the other hand, we have no post-mating isolation patterns detected on this system. They apparently do not show any problem when mating on the wild or on the wild. And so we think that the most important fact in this system is that the isolation is produced by pre-mating mechanisms. We have some previous knowledge on some draft Ohmic technologies, more useful for modest group like us, which have no so big machines like 454 and similar, but which could be quite useful from time to time to get some nice information. For example, we have got some comparison between the differentiation on AFLP polymorphisms on the nuclear part of the genome between these two ecotypes, the wave exposed and the crab protected, and they differing about 3% of the polymorphisms, which suggests that the differences are not affected by just a few genes, not perhaps more than, well, we don't know, but many of them. Of course, we also tried some study using the cDNA with the same technique, and we found that they differ in approximately a similar rate, about 4%. This was already published. We already got some estimates of the differentiation between these ecotypes on a proteomic scale, and it was quite larger, more than double of these estimates, which is, I think, more interesting. We already show that proteomic difference and morphological difference between these ecotypes are quite constant independent of the environmental that the ecotypes have experienced, which suggests, of course, we know that for morphology, but suggests also a strong genetic component for proteomic difference. So the technique that I will present today is a small version of the proteomics possibilities, which is called the two-dimensional proteomic analysis by gel electrophoresis, which is based to the possibility to homogenize a tissue or an organism and separate the proteins by charge or mass and produce a two-dimensional map that can be used to compare different biological factors like the two ecotypes and using within treatment replication like in any classical experimental design. So then it's possible to scan the different spots and compare them after some special normalizations approach. You can get a new variable which can be used like a classical variable on classical ANOVA methods as well as multi-test corrections. The experimental designs that we use is shown here. We use different pools of specimens. Each pool was, we used 10 different specimens, so in total we used 240. And we study 3 different factors: ecotype, of crab protected and wave exposed, sex, males and females, and life stage, different age, embryos, stage, juveniles, and adults. To statistical reasons, we need to analyze this data in 2 subsets. In one, we analyze ecotype and sex, and using this data set. And in the other, in a second experimental design, we analyze ecotype and life stage using this data set. I need to mention that we also use six different technical replicates, and the variability obtained as well as the correlation across replicates was as high or as typically is observed in similar studies in the literature. I will summarize first I will focus on the experiment 1, studying ecotype and sex, and I will summarize the results in figures like this, which represents for the different spots that we analyzed, in this case more than 500, the percentage of significant spots for the different factors, of course corrected by multi-test adjustment. We use a 5% significant level here, and we got a quite high percentage of differentiation between ecotypes, more than 30%, 20, sorry, I don't see, 20%, yes. And sex difference were about 5%, and the interaction was quite lower. And when we repeat the analysis on the different significant level, we got approximately the same results, and obviously with a lower percentage, with less power, less percentage of differentiation. And so from now to the end of the talk, I will just focus on the 5% significant level, but we got exactly the same patterns in the other case. So the main result is that we got a high ecotype differentiation, but low sexual differentiation. The first is quite expected if you check just the morphological difference between the ecotypes, and the second is perhaps something unexpected, and we interpret this because we in fact are capturing the proteomic difference mostly based on the muscle tissue. We have some experimental data which support that. If we focus on some details of this data and do some special sub-analysis, for example, oh, excuse me, bad luck. For example, we can focus on the difference between sex and check them by each ecotype separately. We observe a significant difference only in one of the ecotypes, in the exposed one. And the difference between ecotypes, the frequency difference were clearly significant. This has some sense because we know that from a morphological point of view, size difference between sexes in the exposed ecotype are 3 times larger than difference between the other ecotype. We can now focus on the interaction, and we can use a different approach to investigate how works the interaction between these two factors. For example, using every spot, we can check if it was significant for one factor and at the same time was significant or not for the other. And so then you can build a typical contingency table like that, and we can use typical sexual isolation index and typical significance by maximum likelihood test and things like that. So the results are quite clear and show both approaches that we have no random distribution of significance across factors, and the association was quite high. The maximum was 1. So this suggests that proteins involved typically in both sexual and ecotype differentiation. This pattern could suggest 2 different processes. For example, could be caused by pleiotropy if the same spot was affecting 2 different tribes simultaneously, but also could be advances a different hypothesis that has been used in other organisms and other contexts. That is the case that some male-specific genes show higher evolutionary rates. In the second experiment, we focus on ecotype and life stage effects, and the results were different. We got more differentiation between ecotypes as expected of using more replicates and so having more statistical power. And we also got quite huge differentiation across life stages and proportionally also more interaction. Interestingly, we know that the life stage effect is mostly caused by hybrids, and excluding hybrids from the analysis, you get 1/3 of the differentiation observed before. Checking what's happened now on the ecotype difference and studying them by each age separately, we got some interesting result, perhaps unexpected, that we found is more or less the same differentiation across ecotypes during aging, both comparing embryos and females. Of course, the percentage of differentiation decreased because in these comparisons the statistical power also decreased. So we think that this suggests that probably genes affecting ecotype difference are switched on quite early during development. We can now check what happened with the life stage difference, but for the different ecotypes and sex separately. We can use adults separately with males and females, and we got the same pattern, but it was significant only in females. But the pattern suggests that the exposed ecotype show less differentiation, and that was significant, than the protected ecotype. And interestingly, this is something that can be shown also by morphology, but nobody have never realized of that. If you, well, the view is not too clear, but if you check the shape of this shell, it's more similar to this shape and to this one than this one to this one. So that can be shown by geometric morphometries and classical methods, but it's something that can be shown by both approaches. And we think that this suggests perhaps a mechanism of pseudomorphosis or neoteny affecting the evolution of the SU ecotype regarding the other ecotype. We could also study the interaction following the same approach than before, but checking the association between life stage and ecotype, and at the same time life stage with sex. And in that case, we only got significant results with ecotype and life stage, suggesting again that ecotype has an effect by itself, but not sex. So perhaps that means that the pleiotropy mechanism is more important, or perhaps other possibilities that we have not think about. Very briefly, I want to mention that we got some identifications in the study. Well, these are more or less preliminary results yet, and these are some of the proteins that we could identify. And the color scores means if they were biased in our study, being more expressed in the crab ecotype or in the wave ecotype, or following similar expression. In those cases that you cannot see any square, that means that those spots could not be used in the analysis, because these methods sometimes need to retire different kind of spots for practical reasons. So, I can advance some preliminary overview on patterns and perhaps on process as well. First is that adaptation is putatively responsible of a shift in expression patterns between two other traits, sexes and life stage. And second, one hypothesis that we can advance that will be able to be tested in the future with different methods is that the wave exposed ecotype may have evolved from an ancestral protected form, perhaps by retaining juvenile stage, like we observe in gene expression and morphology. And just finally, a suggestion for all of you, those interested in gene expression should think on the possibility to also apply a proteomic study on your favorite organism. Thank you.
Thank you very much, Emilio. We have time for a few questions. Luis, we have the microphone.
So I guess the potential drawback of it to the migration is the difficulty of getting the identity of the proteins that make the difference between the
There is some other related technologies like shotgun proteomics, for example, that could be used coupled with a known part of the genome of the organism. In that case, it's more easy to at the same time that you estimate quantitative difference, check if to identify those proteins. But of course, you need some advancing working on genomic on these organisms.